Okay, can somebody confirm you can hear me?
Confirming.
Thank you. All right, before we begin, are there any questions? I suppose I should show you something. Let me see if I can get it. I gotta get the light on it here. Can you guys see the monitor here? Anyone?
Yes, the one you're holding?
Yeah. All right. So to do the scan for the environment of your computer, you need to probably move your monitor or your laptop, and you got to point it down. Hopefully you can see I'm doing that now. And then scan the area where your computer is sitting, which I know you can't see that because I can't show you that. And then scan it to the right all the way until the edge of the table and scan it to the left all the way to the end of the table. And if you can't tell where you're scanning, which a lot of you will have that trouble, you want to scan more than is necessary, meaning you can pick it up, scan before where your table is, meaning where your computer is, scan the whole area, then scan where you think your computer is and scan the whole area, and then scan behind where your computer is and scan the whole area. And by the whole area, I want to be able to see the edge of the table or desk that your computer is on, which would be right here. I don't know if you can see that. And then you probably can't see this, but it's over there.
We can't see you.
Yeah, we can't see you, and also you cut out, and I didn't hear the ending of what you were describing when doing the computer scan. I
Oh,
already
you
did
mean
that.
my voice cut out?
Yeah, your voice cut out.
Oh gosh. Let me do that again then. So what you want to do is I'm going to pretend this is the monitor.
We still can't see you, just so you're aware.
You
I
can't
think
see
it's
me.
your background. Your background's like trying to
I
edit
can't
you.
turn off the background easily. So um, let me just say, can you see this now?
Yes.
Usually if
Yeah.
it's in front of my face, you can see it.
Yes.
Okay. So you want to bend it down so you can see before where your computer is. You might be showing me the floor, I don't know. And then just move it to the left so I can see the edge of the table or the desk, wherever your laptop is or your computer is. And then scan it to the right. You can do it slowly too. And so I can see the edge of the table there. And then move it to where you think your laptop is. So I'm getting that area. And then move it so I can see left of where your laptop is until the edge of the desk, and then move it towards the right so I can see the scan all the way to the right where your edge of the table or desk is. And like I said, um, this is especially if you can't see where you're scanning, and then scan it behind where your computer is, scanning it to the left and to the right as well. And that way, I will definitely see all of the area and you'll get full credit. If you don't show me your complete environment, you may get 0 points. And it's true, I think the first couple of scans it's only worth 1 point, but later on it'll be worth 3 points. Any question about any of that? So you do need to scan your environment. That's what Respondus Monitor is doing. And then it's recording you to see if you will be doing anything like using AI or opening up a textbook. So I can watch the entire video if I need to. Now, hopefully, I won't need to, and I'll just take a look at your scan. And if everything's good with the scan, everything's good. Any questions about any of that?
I have a question.
Uh huh.
If we've already submitted the quizzes, like the anti-plagiarism quiz, and included the environmental scan and haven't heard anything, does that mean it's likely fine?
I haven't graded it yet, so I
Okay.
don't know the answer to that. Meaning I
Okay.
haven't looked at the scan. If you think you didn't do an entire scan, you should do the quiz again. Okay? If you know you didn't do the entire scan, you should do the quiz again while you still can.
So you just want to see like the table area where the computer is, you know? Like I scanned the table with my printer on it where my laptop sits, but then I also scanned like the whole room because I'm in like an office chair and so I can spin in a circle. And I showed
Yeah,
you like the whole room I'm in,
more
including the
is
laptop
always
table.
better, okay? That way I will see your complete environment. But like I said, I want to see all the area around your computer. So that's all that is required, all the area around your computer. But if you show more, there's no problem with that.
Okay.
All right, I don't have it marked where the heck we left off. I remember talking about a nitrogen cycle. Let me see if I've got a two here. There it is. Oh, there it is. Yeah, it's the nitrogen cycles where we left off exactly. I know you can't see the screen here. Just let me save that and then I will share my screen here. There it is. They updated the Zoom, and so now I've got to click several buttons to get things done, including starting the session. That's why I was a little late today. I started Zoom and then forgot to start clicking the other buttons and was wondering where everyone was, and then realized, I haven't really started Zoom completely because there's like it's instead of being one click to start Zoom, it's now like 3 or 4 clicks. And all right, so we had microbes in the nitrogen cycle last time. Any question about any of that? You can see my screen, is that correct? All right, so microbes are also important in making soil. There are rock-eating microbes. Some microbes actually obtain nutrients from rocks and they break down the rocks, leading to the formation of soil. Now, do understand that microbes do not make the majority of the soil. Most soil is made by erosion from the wind or from water, but soil making is actually one important thing that microbes do, and they do make a significant amount of soil. It's something like around 20% soil is made by microbes. Microbes are also important in the level of oxygen in the atmosphere. Let me blow this slide up so you can see it so better. If you look at the level of oxygen over the Earth's history, you'll find that initially in the early Earth's history, the oxygen level was 0. And then the oxygen level about 2.7 billion years to 1.8 billion years ago, the oxygen level started climbing in the Earth's atmosphere, getting up to about 5% from close to 0%. And over time, the oxygen level has increased until present day, the oxygen level is around 21%. What happened right here to change the oxygen level between 0% and up to 5%? What happened was microorganisms began photosynthesizing, putting out oxygen as a waste product, and where that oxygen went was into the air, building up the Earth's oxygen level. Once the oxygen level built up to about 5%, oxygen in the Earth's atmosphere could then make ozone, which then protected the land from the UV light. And then green plants could evolve on the land, and green plants and microbes now make up oxygen, brought the oxygen level to 21%. Green plants actually only made about half of that oxygen level, meaning half of that 21%. The other half has come from microorganisms, such as cyanobacteria. Any questions about the importance of microbes to the oxygen level in the Earth's atmosphere? You couldn't have had green plants evolving if the UV light wasn't blocked, and then green plants could start colonizing the land. And here we're looking at a sewage treatment plant. Humans are responsible for generating the waste, right? Like when you flush your toilets and wash something down your sink, you generate the waste. And it comes in these human sewage treatment plants, which humans have made. So humans are breaking it down, right? Anyone object? Somebody should be objecting, and that is humans do not break down the waste in this human sewage treatment plant. Which actually is breaking down the waste are microorganisms, which are growing in the water here, which break down the waste that comes out of your sinks and your toilets. Any question about any of that? So it's the microorganisms which are recycling the nutrients in the sewage treatment plant, and they just break it down into water and carbon dioxide. Microorganisms are organisms which are too small to be seen with the unaided eye. A microbe is a microorganism, and the way your textbook uses these two terms, the textbook uses it similar. I will be a little bit different, and that is an organism is something that is a cell, an organism which is too small to be seen by the unaided eye. A microbe, on the other hand, is all of those microorganisms, but it is also a viruses and a prion. Viruses and prions are special because they are not organisms. They do not have cells. So microorganisms include, obviously, viruses, or I should say microbes include viruses and prions, as well as most algae, all single-celled algae, protozoans, yeast and molds, they're microbes, single-celled organisms, archaea, and bacteria. So all bacteria and archaea are something that would be similar to a single-celled organism, and we call them a microorganism. Like I said, viruses and prions don't have cells, so I don't call them organisms, but your textbook does. So you have to kind of learn it both ways, I guess. I will never test you on the difference here, but so a microbe is a microorganism. You should know that microorganisms are almost everywhere, and we call that the ubiquity of microorganisms. Wherever you go on the Earth's surface, you're likely to find microorganisms. They do play beneficial roles in numerous processes, such as the making of oxygen, the breaking down of waste, and obviously the nitrogen cycle. We'll talk about some of the other important roles that they play on the planet in just a little bit. Microorganisms are numerically the most abundant life forms on the planet Earth. Even if you're only talking about bacteria and archaea, they are the most abundant life forms or cells on the planet Earth. So what is a cell? I've already talked about cells. Let's talk about what is a cell. The cell is the basic structural and functional unit of all organisms. A cell is a highly organized compartment bounded by a thin, flexible membrane that we call the plasma membrane or the cell membrane. Those two terms mean the same thing. All cells contain three components: the plasma membrane, cytoplasm, and then their genetic material, their DNA. Here we're looking at some bacteria cells here. Maybe get a little this up. So in this picture here, where would you find microorganisms? Would you find them on the plants in the soil here?
You find them everywhere.
Yeah, almost everywhere. Would you find them in the water?
Yes.
All right, how about in the seawater? We find them in the seawater. And we're talking about like 1 centimeter of seawater.
Yes.
Yes, you do. How about in the air? You find microorganisms in the air?
Yes.
Yes, you do. You can find microorganisms up about a mile in the air. And the air currents just blow microbes around. And you can find them up about a mile in the air. And how about in the soil here? Obviously, we find them in the green plants in the air, but how about in the soil here?
Absolutely.
Yeah, you find microorganisms down about a mile in the soil. Now, obviously, if you have bedrock shortly underneath the surface of the soil, there may not be microbes underneath the bedrock. But in the soil, you'll have microbes down about a mile. And that's true for the ocean, what do you call that? The ocean surface. From the ocean surface, meaning the seafloor, you can go down about a mile in the seafloor and find microorganisms as well. Obviously, you found microorganisms on the cat and the person here on the door. How about in the desert sands of the Sahara? Do you find microorganisms in 1 centimeter of desert sand?
Yes.
Yes, you do. All right, this is a volcanic vent, and that orange stuff is either magma or lava. Do you find microorganisms in the lava or the magma?
Yes.
That's a good guess, but guess again.
No.
It's too
Yeah,
hot
the
for them
answer
to
is
survive.
no. This is way too hot for cellular organisms to survive, and the viruses will be destroyed too because it's too hot for them too. So that's about the only place on the planet Earth you do not find microorganisms, and that's if lava is flowing or magma has reached the surface of the Earth. But you do find it on the ice here, even on the edge of the vent there, because that's not where the lava is. And there you got a picture where you find it everywhere except for right there. Any question about any of that? So you find microorganisms essentially everywhere on the planet Earth, with one exception, that's where lava is actually flowing or existing. So, where is everywhere? That's any environment where microbes are found on the land and in the water. We're talking about in the surface of the water, down about a mile, assuming you're in, what do you call it, sediment, or on the land, down in like a mile in the soil. I'm not counting, obviously, bedrock because, especially if the volcanic vents made the bedrock, and there won't be microbes underneath that, but otherwise down about a mile in the soil, about a mile up in the atmosphere. And you find microbes on all plants and animals. Essentially, wherever liquid water is present, you can find microorganisms. Microbes need to exist between temperatures of a minus 20 degrees Celsius and about 120 degrees Celsius. Remember, at 100 degrees Celsius is the temperature water boils at, and so we can have cells alive higher than the temperature of boiling water, at least at
Is that why
standard.
when you go to the hot springs at Yellowstone, it's all like multicolored and stuff? Is because of all the microbes?
Yeah, usually the color is from the microorganisms. It's true that some minerals could have color, but most of that color is from the microorganisms. And that will be some microbes living in temperatures above 100 degrees, giving the color. And then you have cells going down to minus 20 degrees C, which the cells have antifreeze in them, and there's alive and metabolically active at 20 degrees C. Below 20 degrees C, all cells will freeze. That may not kill them, like bacteria can survive freezing temperatures and then thawing, but only about 50% of the cells can survive that. But my point is, you can have microorganisms living down to a minus 20 degrees Celsius, and that's because they have antifreeze in the cell, and they can be medically active, at least some cells, down to minus 20 degrees C. But below that temperature, the cell will freeze. It may not kill it, but the cell will freeze. And that temperature would be a minus 4 degrees Fahrenheit to about 250 degrees Fahrenheit. Any question about any of that? So how many microorganisms are there? I mentioned that they're the most abundant life forms on the planet Earth. And when we're talking about microorganisms, in this sense, we're only talking about prokaryotes. If I were to say prokaryotes and viruses, the numbers would be 10 times what I'm going to discuss. But in 1 gram of soil, that's about 1 cubic centimeter of soil, you can find 10 to the 8th prokaryote cells. It does depend on the amount of organic matter in the soil and the amount of moisture in the soil. Like in our soil of Western Washington and Oregon, we probably have a little bit more because our soil, at least at the moment, is quite wet. And you're talking about the soil of the forests of Western Oregon and Washington. There's got lots of organic matter in that soil as well. So our soil will be a little bit more, but on average, for most soil, it's about 1 times 10 to the 8th prokaryote cells per gram of soil. Now, if we're talking about desert soil, where the soil has very low organic matter and very low water concentration in the soil, you'll get less cells than this. In fact, the desert soil, especially a really dry desert like the Sahara Desert, will have only about 1 times 10 to the 6 cells per gram of soil. But soil on average about 1 times 10 to the 8th cells per gram of soil. How about in 1 milliliter of ocean water? If you don't know, 1 milliliter is about 1 gram of ocean water. Do you think you have more or less water, cells, than 1 gram of soil? You want to have a guess? You're either yes or no, so you got 50-50 chance here.
Soil, probably.
Soil has more, you're correct. And that is ocean water is actually one of the deserts on the planet Earth. So we actually have less cells in ocean water than we have in soil. How about in 1 gram of human feces? And we'll just say a fresh human feces.
Does salt water affect a prokaryotic cell the way it affects a eukaryotic cell? Like, does it make it swell, or like, does osmosis happen with prokaryotes?
Yes, osmosis happens with prokaryote cells as well, but obviously if the cell is living in the ocean water, then the cell will have high salinity or high hygroscopic pressure in the cell, meaning a lot of sugar or something like that, to keep the water inside the cell.
Okay.
And then to prevent the cell from bursting, the bacteria, or at least most bacteria, have a cell wall around it, and that would prevent water from moving into the cell and causing the cell to burst. And actually, plant cells have a cell wall around them too. And so plant cells can be fine in like a stream or a pond Whereas if you know your red blood cells do not have a cell wall around them, and so you put them in fresh water or distilled water, water will enter the cell and keep entering the cell until the cell membrane bursts, and then the cell will die. All right, 1 gram of human feces has more cells, especially fresh human feces, than 1 gram of soil. On one human being, we have more cells than in 1 gram of soil. In the entire earth, we have many more cells than in 1 gram of soil. And they're astounding numbers. In the ocean water, we have about 1 times 10 to the 6 prokaryote cells per gram of water or 1 milliliter water. And like I said, most soil, about 1 times 10 to the 8th. But well, let's talk about a wet human feces. You'll have about 1 times 10 to the 11th cells per gram of human feces. That human feces is actually about 50% of the wet weight is from microbes in that human feces. I don't know if it's counting water. It probably is, but because the cell has water in it too. Now, if you were to go to the bathroom in the Sahara Desert and the feces were to dry out, you would only find about 1 times 10 to the 6 cells in the human feces, which is what you find in the desert soil, by the way. About 1 times 10 to the 6 prokaryotes per cell per gram of desert soil. On a human, you got about 3.8 to 10 to the 13th cells. The entire Earth, we have 1 nonillion. That's 1 with 30 zeros after it, or 1 times 10 to the 30th. For comparison, the human population is around 8 billion. This number here, much smaller than a nonillion, which is the population of microorganisms, or at least prokaryotes on the planet. Any question about these numbers? Now remember, if we're talking about viruses, you'd have to take this times 10. All right. Microorganisms can cause disease. If they do cause disease, we call them a pathogenic microbe. So a pathogenic microbe is a microorganism that can cause disease. You should come to realize that most microorganisms do not cause disease. Only a small percentage of all microorganisms are pathogenic or disease causing. Any question about any of that? When we're talking about diseases, it's important to distinguish between an infectious disease and an inherited disease. An infectious disease is a disease caused by a microorganism or a worm, and an inherited disease is one that a person is born with. So 2 types of diseases. Can anyone give me a name of an infectious disease?
MRSA.
Oh yeah, MRSA or MRSA, causing generally a skin disease, or if it gets in the blood, causing a blood infection. I mean, MRSA can get in other parts of the body too, but usually the skin. Can anyone name another infectious disease?
AIDS?
Say again?
AIDS?
Yeah, HIV causing AIDS would be another infectious disease. Can anyone name an inherited disease? Some disease that somebody is born with? Well, how about sickle cell anemia? The child who has sickle cell was born with 2 sickle cell alleles, causing the child to have sickle cell anemia.
I was gonna say rickets, but that's caused by a deficiency. I don't think that's inherited.
Yeah, I think rickets is caused by the environment. You get a vitamin deficiency, which I think is vitamin C deficiency.
Could
But
herpes be an inherited and a infectious disease?
herpes, no, it's an infectious disease. Even if you get it at birth, you got it probably from your mother during birth, and so that's an infectious disease. Unless you're talking about oral herpes or cold sores, in which case you get it mostly from your parents, presumably from when they're kissing you, but it could be maybe when you're nursing as well. Um, but it's an infectious disease as well, caused by a microorganism, in this case, a virus, herpes, but
I
definitely
think
a
like an
micro.
enlarged heart is like an inherited disease caused by genes or alleles on genes or something.
That one could be caused by inherited, meaning that you got the genes to cause it, but it could also be caused by your environment. Like my mother got an enlarged heart as she was aging, so she never was born with it. It happened just
Later in
from
life.
environmental stress, I'm
An
guessing.
enlarged heart is a genetic disorder in my family, so that's why I said that. But
Yeah, like I said, that one can be inherited. I didn't say it wasn't. I just said it can be and caused by environmental stress as well. All
yeah, I
right.
don't, yeah.
I'm not going to talk about diseases caused by the environment, but a good one would be tennis elbow. Somebody who likes playing tennis after they've done it for a long period of time, their elbow doesn't quite work as well as it used to. And it's from the stress on the elbow from playing tennis for so many years. That one is clearly an environmental um, disease, meaning it's caused by the environment. It's not really caused by the genetics, nor is it an infectious disease caused by a microorganism. But there are many diseases like diabetes, which actually has a genetic component. It has an environmental component, and well, it may have an infectious component. There's some evidence that some infections can at least lead to the increased risk of getting diabetes. So there's some diseases which have all three factors. And I'm not going to really talk about environmental factors much in this class at all. But we will talk specifically categorizing diseases into inherited diseases, one that a person is born with, and infectious diseases, which we'll talk a lot about in this class, a disease caused by a microbe. All right, here we're looking at some microorganisms. How small are they? Let me blow this slide up. Most bacteria are around this size, meaning about 1 micrometer long. This one's even a little less in width, and that one's a little longer. It's a little larger than the average bacteria. This is a fungus, maybe penicillin, and there's its shape there. It's much bigger than a bacteria. And each one of these are a cell here. That's probably a white blood cell, also a microorganism, something you need to use a microscope to see. There's size there, 10 micrometers. So that's, well, actually, that's close to 50 micrometers. So that would be like 50 times that cell. And then here we actually do have a microbe because you need a microscope to see it. But actually, each cell is one of these dots here. And this is the parent colony. So this microbe, its Volvox, is growing in a colony. And there's the parent colony. This parent colony has made 6 baby colonies right here. And the parent colony will open up, and then the babies will come out and be carried by whatever this is, the current, to wherever they go. And then the baby carrot colony will become its own parent colony. So there are some microbes which are more than a single cell, and Volvox is one where it's colonial, and I don't know how many cells are in there, but it's a large number in just the parent colony. Obviously, probably twice that number we put in the baby colonies. Most bacteria are single individual cells. There are some bacteria that do grow in colonies. However, we do call them single-celled organisms because if you break off one cell from the bacteria colony, that one cell can go on and form its own colonies, meaning one colony, a single cell can break off and then go on and form its own colony. And so we do call these bacteria single-celled organisms, even if they're growing in a colony, because you break off one cell, it can form its own colony. Any question about any of that? I don't know about Volvox. I don't think if you break off one cell, it can form its own colony. So I don't think that one would be a single-celled organism. And then there's another microbe. This looks like a helper T cell, the big one there. And then these red dots here, those are actually HIV. And I think they're coming out of this cell, but they may be going infecting the cell. So that'd be HIV viruses infecting or coming out of this cell. Any question about any of that? You can see that the microorganisms have different sizes and shapes, and that some of them can be colonial. All right, let's talk about some of the beneficial roles that microorganisms play. We already mentioned that they're important in decomposing organic waste. This is especially true in the forest. When the tree dies and falls down in the forest, it's mostly the microorganisms that decompose the tree, making the nutrients inside the tree available to other organisms, including other trees, but other microbes and that. And it is true that there are mushrooms that help decompose the organic waste as well. But all decomposers are either a mushroom, a fungi, or a bacteria or archaea. So most decomposers are microorganisms. And even some fungi are microorganisms. But a mushroom is a colonial organism to make the mushroom you can see. And so that would not be a microorganism. That would be a mushroom, not a plant, but a fungi. There are microorganisms which are producers in the ecosystem, such as the photosynthetic microorganisms. And these microbes engage in photosynthesis, the same as green plants. So they're important on the ecosystem because we need photosynthetic organisms to make food for us or for animals, as well as for the green plant or the green microbe. And then there are chemosynthetic microorganisms, and they can make their own food. So they are a producer in the ecosystem, but they use chemicals, not sunlight, to get their energy. And we'll talk a little bit about them. They're the chemosynthetic microbes. Only microbes can be chemosynthetic. There are no eukaryote cells which are chemosynthetic, so there's no plant or animal that's chemosynthetic. Only microbes. We've already mentioned that microorganisms are involved in the nitrogen cycle, and you would not get the nitrogen cycle unless you had microbes converting the nitrogen from one form in the cycle to another. Microorganisms Microbes are important in making industrial chemicals for humans, such as ethanol or acetone. The ethanol that you get in gasoline is made by a microorganism. And it's true that you probably get the sugar from corn, but it's the microorganism which makes the ethanol. It's not the corn plant itself. We use microorganisms to make insecticides. We'll talk a little bit more about that. We use microorganisms to make fermented food, such as vinegar, cheese. To my knowledge, all cheese, maybe not all cheese, but most cheese is made by a microorganism fermenting the milk into cheese. A leavened bread uses a microorganism to leaven the bread or put air in the bread. And then beer, as well as wine, fermented foods that we make using microbes. Yeast ferment the hops and the, what is it, grain barley, whatever they make beer out of. I should just talk about wine. You make most wine out of grape juice, and it's the yeast which ferments the grape juice into the wine. We use microorganisms to make products used in manufacturing, such as cellulase. We use microorganisms to make antibiotics, such as penicillin G. The first member of the penicillin family was made by a microbe. The penicillium mold, we'll talk a little bit about that. And then we use microorganisms to make other products used in medicine. Today, all insulin that we give to people is a human insulin, but the human insulin gene was taken out of a person and put into a microorganism, and then we grow up the microbe, and the microbe makes the human insulin, and then we purify the human insulin, and then make it available to people. My father was a diabetic, and he had to take insulin shots, and at the very start of it, they actually purified insulin from people, because I guess that was the first way they got it. And that wasn't very efficient because not too many people wanted to have their blood drained, and then they pull out the insulin from it and purify it and then package it and then give it to people like my father. It would be very inefficient. So what they did was they got it out of sheep, harvested the sheep to get the insulin, purified the insulin, gave it to my father, and then later he got human insulin again, but it wasn't made by a human. It was made by probably a bacteria that they put the human insulin gene in. Any question about any of that? All right, let's talk a little bit about biotechnology and genetic engineering. Biotechnology is the use of microbes and organisms to produce food and products for human use. Biotechnology is actually centuries old. An example of biotechnology is making kimchi or, oh, what do you call that? Coleslaw, not coleslaw, but yeah, coleslaw. Fermented cabbage to make coleslaw. Biotechnology to make a human product for human use. Centuries old. Most people think of biotechnology as genetic engineering, a new technique of biotechnology where you take out the genes of one organism, put it into another organism, and then grow up the gene of interest. We can use genetic engineering to make a variety of proteins from bacteria and fungi, such as making a vaccine in a bacteria or a fungi, probably a bacteria. We can use genetic engineering as gene therapy, like a child who is born with a genetic disease. You can use gene therapy on the child and then make their body able to act like normal for at least a few months. we'll talk a little bit about that. But usually what they do is they put the correct human gene into a vector and put that vector into the child's immune system cells, like their T cells and their B cells, grow them up, and then give the child those cells. And then a child which was born with a defective gene, let's say a child unable to make antibodies, the child is now able to make antibodies for at least a few months. The bad news about gene therapy is it has to be repeated every couple of months. So the child has to have this therapy every few months for each year of the child's life. But the good news is instead of having a child who isn't able to make antibodies, for example, the child now can live like a normal individual, at least for a few months. And they, at last I heard, decided to keep this arrangement because somebody did invent it so that you're putting it into the child permanently. But that tended to cause cancer later on in life. And so that process has been stopped. And so they're doing it where the child has to have the therapy every few months. Now, there is an exception, and that is, I think, if you know the child is going to be born with a defective gene, you can pull out the cell or fertilize it in vitro and then correct it while it's one cell, and then implant that cell in the mother, and that child can then be born with gene therapy. But like I said, that's the very first cell stage of the child. So I think you can correct it now at the very first stage. And I think that doesn't cause cancer. But if you have the child is already born, you need to correct the cells and then put them in the cells every few months. Any question about gene therapy? And then we can make genetically modified bacteria, such as modifying the bacteria to prevent crops from being eaten by insects, or genetically modify the bacteria to then spray the plants with the bacteria and then prevent freezing. I'm sure you've all heard about genetically modified tomatoes, the was it flavor savor tomato, which is very hard to find now in the supermarket. Apparently people didn't want to buy it, so people stopped carrying it. But we can make genetically modified crops like the flavor savor tomato, and also genetically modified bacteria. Any question about any of that? All right. Let's go on and talk a little bit about microbial ecology. Bacteria are important for recycling nitrogen in the earth, planet Earth. They're also important in recycling carbon on the planet Earth, nutrients as well as carbon. When something dies, the microbes as well as the mushrooms will break down the dead tissue, releasing the nutrients and the carbon, so these can recycle on the planet Earth and recycle in the ecosystem. Microbes are also important in the sulfur cycle, the phosphorus cycle, and that way when an organism dies, the sulfur can be recycled and then made available for future plants and animals. The sulfur cycle and the phosphorus cycle happen similar to the nitrogen cycle. So let's go back to the nitrogen cycle. It's just that in the sulfur cycle, there are not microbes making each step of the cycle essentially work. Not each step of the cycle works in the nitrogen cycle either, like nitrates going to green plants, and then green plants converting the nitrate into an organic compound in like protein or something like that. But all the other steps are a microorganism which are having the cycle work. In the sulfur cycle, there are a few steps of the sulfur cycle which do not use a microorganism or may not use a microorganism, such as sulfur leaching from rocks, entering then the sulfur cycle. But the sulfur cycle and the phosphorus cycle do use microbes to help the cycle work. And then recycle those nutrients in the ecosystem. These are some examples of the microbial-mediated biochemical cycles that support life on Earth. These microbial nutrient cycling systems were first discovered by Winogretsky and Bajorink, and they first saw it in, I think, the nitrogen cycle and the sulfur cycle. And the reason why they were able to discover this was because they were unlike most other microbiologists, and that is they didn't study one microbe purified. They studied a whole ecosystem composing many microorganisms. And to get the nitrogen cycle and the sulfur cycle, you need to study many different microorganisms to find the microbial mediated biochemical cycle, meaning you need all the organisms which are involving one source of nitrogen converting to another, and then another microbe converting that source of nitrogen to another. So they were unique, and that is they studied many different microbes, and that's why they were able to find the microbial mediated biochemical cycles. Any question about any of that? Now you should know that Winogretsky and Bajorink discovered the microbial mediated biochemical cycle supporting life on Earth, but you do not need to know that it was from 1880 to 1892 that they discovered these different cycles. You don't need to know the year. Any question about any of that? All right, microbes are important to human welfare, and that is microbes can be used in bioremediation. Bioremediation means life, and to remedy, where we use living organisms to deal with an environmental problem. Such as this oil spill here, these gentlemen are spraying microbes on the oil, so the microbes will digest the oil and then break it down into CO2 and water, removing the oil. So that's bioremediation. We can use bioremediation to break down sewage, but we also use bacteria to break down pollutants, like oil here, an oil spill being a pollutant, or also mercury. Now, when we treat mercury with an organism like a bacteria, we make it a less toxic form of mercury. You just don't break down the mercury because mercury is an element. It's an atom. But we can make it less toxic. Any question about any of that? We can also use microbes for a biological insecticide. Some microbes are pathogenic to insects, and they're an alternative to chemical pesticides. Chemical pesticides are toxic to humans, and long-term exposure causes, I don't know, various side effects. If you were to use a biological insecticide, it's less toxic to humans. Some of the biological insecticides are not toxic to human at all, like Bacillus thuringiensis is an infection which is fatal to many insects, but this bacteria is harmless to humans, animals, and plants. And there you have a bag of Bacillus thuringiensis. My father actually bought this in the 1960s and put it on the apple trees. However, he was too lazy to repeat it about every 2 weeks, which is what you have to do when you're spraying apples with a chemical pesticide. You have to spray it on every 2 weeks once the apples start to form until close to when the apple is getting ready to pick. You need to spray the apples every 2 weeks. So my father only treated the apple tree with Bacillus thuringiensis one time, and he was in Western Oregon. And so obviously once it rained, the Bacillus thuringiensis washed off the apples. So obviously the apples did get worms, but they did get fewer worms than if you hadn't treated them at all with Bacillus thuringiensis. For this to work, like I said, you need to treat the apples about every 2 weeks. And you also need to treat the apples after the rain, because the rain will wash it off the apples. Any question about using biological insecticides? All right, any questions on anything we've discussed so far? If not, we're going to be changing gears a little bit and talking about the naming and the classifying of microorganisms. All scientific names, as well as the first classifying of organisms, happened by this gentleman here, who established a system of scientific nomenclature. He also established a system of classifying. Anyone know his name, who this is?
So Linnaeus.
Yeah, this is Carolus Linnaeus, which your textbook calls Carl Linnaeus, meaning he liked Latin so much he Latinized his name. I think his name, I think he was Swedish. In Swedish, his name would be something like Carl Linné, and he liked Latin so much he Latinized his name, and so most people call him Carolus Linnaeus, but our textbook calls him Carl Linnaeus, meaning they only changed his last name. They didn't change his first name. The scientific nomenclature has each species having two names, a genus name and the specific epithet name. For example, for humans, our scientific name is Homo sapiens. Homo being the genus, and then sapiens being the specific epithet. But to name the specific species, you need both. So a binomial nomenclature is what this is also called, the scientific nomenclature or the binomial nomenclature, because each species has two names. Any question about any of that? Let me state that he also started the classification system. His nomenclature system is so good, we're still using it today. So everyone agreed his scientific naming system was very good, and we still use it today. His classifying of organisms is not used today. He classified all organisms into plants and animals. And as you know, there are organisms which are not plants, which are not organisms, are not animals, like there's fungi, and there's microorganisms. All of those are not really a plant or an animal.
What was the guy's name that started this?
Carolus Linnaeus, or Carl Linnaeus is what your textbook calls him. His specific name is Carl Linné, but nobody uses that. This is his real Swedish name. Most people call him Carolus Linnaeus because he changed his name to that. When you're using the scientific nomenclature called the binomial nomenclature, all names are either italicized or underlined. So Homo sapien here, I've got it italicized because I can type it. But if I were to write it on the board, I would write Homo sapiens and then underline it to mean it's a scientific name. Any question about that? The genus name is always capitalized, so the H in Homo is always capitalized, and the specific epithet is always lowercase. All names are Latinized and they're used worldwide. What does that mean? Homo sapien will work wherever you are in the world because everybody knows that Homo sapien is the word for any language for humans, meaning it's used worldwide. Any question about any of that? The name can be descriptive or honor a scientist, like Homo sapiens means wise man that walks on 2 feet. Escherichia coli is both descriptive. Coli, you can find it in the colon, and then it is honoring a scientist, one of the first microbiologists. He may be the first person found E. coli, I'm not sure, but Dr. Esther. Staphylococcus aureus is descriptive. It literally means golden aureus, balls, coccus, that grow in a grape cluster. Staphylo means grows like grapes. Any question about any of that? All right. After first use, the scientific name may be abbreviated with the first letter of the genus and then the specific epithet spelled out. So the first time you use Staphylococcus aureus, you have to call it or write Staphylococcus aureus. But after the first use, all later uses, you can go S period aureus. You should go Escherichia coli the first time, and then all later times go E. coli. However, E. coli is so well known that even among scientists, oftentimes it's just called E. coli the first time it's used. So E. coli is sort of an exception, even among scientists, and that is, it's often just E. coli, and strictly speaking, by the rules, it should be the first time Escherichia coli. Both Staphylococcus aureus and E. coli are found in the human body. S. aureus is actually found in about 20% of Americans growing on the skin, about 50% of Americans growing in the, the upper throat and the nasal passages. Staphylococcus aureus, though, is a human pathogen, and it can cause skin disease. So if you're the unlucky person who has it on your skin, meaning the 20% who has it on your skin, you probably are more likely to get a skin boil or a skin disease from Staphylococcus aureus. Fortunately, most of us, mainly 80% of us, do not have Staphylococcus aureus growing on our skin. And for whatever reason, it doesn't cause usually nasal infections, but it does tend to cause skin infections. E. coli is found in everyone's gut, in the colon and the large intestine. Actually, E. coli is something that helps us digest our food. All right, any question about any of that? All right, so we've talked about the naming of organisms. Now let's talk about the classifying of organisms. As I stated, this was began by Carolus Linnaeus, but we don't use his classification system anymore because he classified everything into plants or animals. And before we can talk about the classification of microorganisms, we need to take a look at the classification of all living organisms on the planet Earth. We divide all living organisms into the very first classification, the three-domain system, where you can classify all living things into a domain. The domain bacteria, the domain archaea, also called the archaebacteria, or the domain eukarya. The domain is actually defined. It is based on the genetic data of the organism. So if we find a new organism and you want to know what domain it goes into, you just need to sequence the organism's DNA or their genome, and then you can figure out which domain it is just by seeing what its sequence of DNA is similar to, meaning all bacteria have their DNA more similar to every other member of bacteria than any other organism in any other domain. Is that clear to everyone? So if you sequence all of the bacteria, every one of them will have their DNA more similar to a member of bacteria than to a member of any other domain.
So they're classified by their DNA.
Yes. Now, usually they classify organisms based on their, I think it's ribosomal RNA, but maybe the tRNA as well, in which case you could classify it on the RNA. And the reason why they use ribosomal RNA is because all organisms have the ribosomal RNA genes. If you were to look at the genes causing the human eye color, for example, well, many organisms do not have eyes, and so there would be no gene to compare it to. So that's why we tend to look at the ribosomal RNA genes for classifying organisms. But it doesn't matter where you look at because all of the DNA of, I don't know, Staphylococcus aureus will be more similar to the DNA of E. coli than to any other DNA in an organism in archaea or an organism in eukarya.
So will we be focusing on mostly bacteria in this class then, or are we going over all three?
Today, we're going to go over all three, and it is true that we do focus on organisms in this class which cause disease, which are largely the bacteria, but there are some eukaryotes which cause disease, like Candida albicans can cause a skin infection, a yeast bloom on the skin or yeast infection on the skin, and then there's some parasitic worms which also cause disease, and those are eukaryotes. So these are the organisms we're going to focus in for most of this class. But today we will talk about all three domains. Okay? All right, the second domain is the archaea. These bacteria are prokaryotes, and it came as a surprise when people started sequencing the DNA that the archaea were in a different domain than the bacteria. They're both prokaryotes, and we thought they were similar, and they're actually not similar. In fact, the archaea are slightly more similar to the eukaryotes than they are to the bacteria, and that's because they had a common ancestor, the archaea and the eukarya, for a longer time than the bacteria and the archaea, or the bacteria and the eukarya, meaning the bacteria separated first, the archaea and the eukarya continued for a short time with the common ancestor, and then broke into two different domains. So it's very slight that the archaea are slightly closer to the eukarya than the bacteria are to the eukarya, or the archaea are to the bacteria, or whatever. Any question about that? So actually, these two are closer together than these two, but very, very slightly, because the archaea and the eukarya did branch a long time ago. So they are their own complete domains. They're not a similar domain. They are separate. All right. Once we've separated organisms into a domain, we can separate organisms into a kingdom. The kingdom is not a defined group. There is no definition for what a kingdom is. There are also arguments among scientists for how many kingdoms there are. Some people say there's only 3 kingdoms, the same as the domains, and so each domain is a kingdom as well, and just give them the same name. Other people say, no, there's 5 different kingdoms. Other people say there's 6 different kingdoms. Other people say there's 15 different kingdoms, 20 different kingdoms, higher order number kingdoms, I don't know, like 30, 40, 50 different kingdoms. It just depends how you define the kingdom, I guess. For us, we're going to separate the kingdoms into 6 kingdoms. And that's mainly because that's what most scientists tend to follow. And it's the last kingdom approach, which goes back now almost 20 years, when people separated life into 6 kingdoms. And that includes the kingdom bacteria, the kingdom archaea, and these two are both a kingdom and a domain. Sorry if that's confusing, it's just the way it is. And then the domain eukarya is separated into 4 different kingdoms. Kingdom plantae, the plants, kingdom animalia, the animals, kingdom fungi, the fungus, and then kingdom protista, the protists.
What's a protist?
A protist is a eukaryote, which is not a plant, not an animal, and not a fungi. So the kingdom protists, everyone always knew was not a monophyletic group. It was just the leftover eukaryotes, tending to be the more simple eukaryotes that people put in the kingdom protista. The plants are a monophyletic group. In fact, there's a definition of what a plant is, but we're not going to go into that. But the point is the plants will be more similar in their DNA sequence to other plants than they are in the DNA to an animal. The protista, we don't have that luxury because some of the protista are further away from other protista, and probably some of the protista are closer to like the humans than they are to other protists. So the protista are not a monophyletic group, and that was always understood. It's just the eukaryotes, which it's not plant, not an animal, not a fungi. And we're using this as the classification system, mainly because it was the last classification system that was generally approved by biologists, as well as to get the higher number of kingdoms, like from the 9 kingdom or the 15 kingdom, you simply divide the kingdom protista into more than one kingdom. Once you get 20 kingdoms or higher, you then start dividing the plants into 2 kingdoms and the archaea into like 3 kingdoms and the bacteria into different kingdoms as well. But we won't talk about any of that. So the only one you need to really know is the 6 kingdom approach, where we have kingdom Bacteria, which is also a domain, kingdom Archaea, which is also a domain, and then domain Eukarya, which we separate into plants, animals, fungi, and the protists. Any question about any of that? All right. Now your textbook says the bacteria have a cell type, which is prokaryotes, and I've actually already talked about that word. And the archaea have a cell type that they're calling prokaryotes, meaning you only find prokaryote cells in the archaea. And the eukaryotes have only eukaryote cells. Other people can call this a superkingdom, the superkingdom prokaryotes and the superkingdom eukaryotes. And that is actually above the domain system where you have all living life things, and then you separate into the superkingdom prokaryotes, the superkingdom eukaryotes. And then the superkingdom prokaryotes are separated into two domains, but the eukaryotes is only one domain. Any question about any of that? All right. If there's no questions, I'm going to end it here. And I'll see you at 6:30 for the lab. Any questions? All right. Goodbye.